and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self-protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell.
Hey, they call that a self-protein. What's another word for self-protein? MHC. So that's what that little square would be. Presenting in.
Helper T cells are key components in the immune response. There are many kinds of helper T cells, each with a unique kind of T cell receptor. Each receptor binds and responds to a particular self-non-self combination of antigen and self-protein, making the immune response highly specific. Binding of the T cell receptor to a particular antigen and self-protein activates the T cell. A chemical signal, interleukin-1, enhances this activation. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activate specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen.
Okay. Hopefully that was illuminating. I'm going to try to kill the T cells up there. I'm going to jump to, that's all I'm going to show. Okay, so I'm going to jump to diseases. I want to point out, guys, that I have simplified this greatly, and there are a lot of other types of T cells, and I'll mention them, but as far as the test is concerned, I'm focusing on those three, but you should be aware there are also suppressor T cells, and they kind of back off on your immune system a little bit, slow down the immune fight after infection. There's also the RAGE type hypersensitivity T cells. It's a subclass of helper T cell, and they're really involved more with allergic reactions, things that like poison metal reactions, chemicals, also. So, and then there's gamma delta T cells. About 10% of all T cells, they live mostly in the intestine, and they're activated by specialized receptors, and I won't get into that either. But so the thing is, you're going to find some other weird stuff when we're learning more and more and more about the immune system. And you have to think about it, there's not only the Army and the Navy and the Marines, right? There's also the Air Force, and then you've got sub-branches within that and branches. So you can just imagine that there's all kinds of different variations on this thing. And we're just really scratching the surface of understanding how the body's on this. This is why it's so huge in the medical field, because as we're learning more, we can apply it to diseases. So, and one of these diseases that's been a really tough one to conquer is cancer. So we know that, right, where does cancer come from? Outer space? No, it's you. I'm sorry, but we all have our little faults and flaws. Cancer is one of them. And And this is also push-pull because cells have it's something called the Hayflick number. Has anybody ever heard of the Hayflick number? It's basically that cells are designed to only divide maybe about 50 or 100 times in a lifetime, and then they die. And they're programmed to die. And you may say, well, dang, well, if we just turn that off, we could live forever. We could be immortal. We really could. But the problem is that some cells that become immortal are really become these cancer cells. They don't behave like normal. They don't die when they're supposed to, and they keep dividing. So there's sort of a trade-off here about maybe we're supposed to live forever. Maybe cells are not supposed to be some cancer. One of these little offshoots of that. They don't do what they're supposed to. They just keep growing. They don't have any sense of space. They bump into something. Normal cells, if it's crowded, you go into a crowded room, you're not going to just like start pushing against people. You just kind of like give you a little space. These cells don't do that. They crowd in and they just push and they'll keep growing. And they don't even care who's around. And they'll actually physically kind of harm the other cells around them. Like if you think of a brain cancer or tumor, it expands, it tends to going to push it out and smash into the other cell. That does bad things. So they don't know when to stop growing and dividing. What's the process of cell division called? You guys remember that? Mitosis. So really it boils down to understanding mitosis and what regulates mitosis and cell division. Because if you really want to understand cancer, you've got to learn all about the checkpoints in mitosis, what proteins are involved in regulating that, what turns it on, what turns it off. So uncontrolled cell division though makes the tumor, and it can crop up anywhere. It can be liver, lung, breast, brain, you name it. Any cell potentially leave. So fortunately, your own body can sometimes recognize these cancer cells. They're figuring out that there are certain marker proteins, markers for types of cancer, like breast cancer is the most common cancer in women. So wouldn't that be really fantastic if we figured out a way to stop breast cancer? We're working on it right now, and they're finding particular markers of breast cancer, but now they're finding out there's a lot of different types of breast cancer. So it's not like one particular cancer, we're working on all types of breast cancers. So we're right at the cutting edge of that, but that's pretty much the key thing is finding unique proteins or marker proteins on cells that your immune system can recognize. And that's what all sorts of companies in Seattle are doing, and throughout the Northwest. There's a lot of companies in California, East Coast, but there's certain, I'd say Seattle is definitely a hotbed of this kind of research about how to figure out how to stop cancer. So if you look at normal cells, you put them in a dish, that's a petri dish, and the cells are red and they're growing. Normally when they hit the edge of a plate, if you grow them, they could be liver cells, you grow them in a dish, it's 37 degrees C, which is body temperature, nutrients, they will stop. That's called contact inhibition. And you put cancer cells in that same dish, they keep growing because they have nutrients, they have space, they hit the edge, and they don't stop. And they will start to pile up on one another, and they make that little pile, and that little pile is essentially what a tumor is. So what's the difference between the normal cells that know when to stop growing and the cancer cells that do not know when to stop growing? They're both liver cells, but one stops, the other does not. So that's a big deal, this idea of contact inhibition. Somehow cells have a sense of space. Okay, now skin cancer is one of the common forms. I won't really get into this super deep on the testing, but the different things. You look for any splotch on your body, a color change, size change, or rapid growth. They call this the ABCD rule, which pretty much to me is silly because they all mean the same thing. ABCD rule, look for asymmetry. Asymmetry means, is there a weird shape on the edge? Border irregularity. Well, that means the same thing. That means you change the shape on the side. Color changes. Well, yeah, color changes. You go from light to dark usually. Diameter changes. That's a change in the shape. So anything that changes the shape of color, keep an eye out for it. Now, skin cells grow fast, which is why they're very susceptible to cancer. You're supposed to grow fast because you're constantly regenerating your skin. And it's subject to all the chemicals, UV radiation. Basal cell carcinoma is the most common, about 75%. Fortunately, it's not that nasty. It's the least malignant. To malign something is to be bad. you have a cancer that metastasizes, it's going to move. Let's say it starts in your lung, it might move to your liver or your heart or somewhere else, or your intestines. So squamous cell carcinoma, those are the flatter cells. That's a little less common. That's for the stratum spinosum. I don't know if you remember that. They're moderately malignant. Well, you remember that I think there's the 5 little layers of the strata of the epidermis, or the stratified squamous epithelium. That stratum of the cell, or the basal layer, is the growing one. And then the stratum spinosum, there's sort of an area. And then, well, eventually, but there's also melanocytes, which are the pigment-producing cells, which is why the color can change. It doesn't matter if you're black or white or Asian, we all have the same melanocytes, and there's just amount of pigment and they look slightly different. But melanomas are pretty rare. They arise from melanocytes. They're highly malignant, which means they're very bad, and they frequently break apart and spread. So those are ones that you really need to detect early, because you might have about a 50-50 chance of surviving if you detect that early. So these other ones, a lot of people get these little bumps and things, you can remove them. Most of the time they're just cosmetic, but you never know. So you got any kind of weird bump or color change, you should get it checked out. Because if it is a melanoma, you want to know right away. So cancers, not only some cancer, but they could start off as a nodule. This would be a non-malignant tumor, a benign, harmless tumor. They don't look very good, but they don't spread. The dogs get them a lot. Big old lumpy, weird tumors on them. They aren't necessarily going to kill them, but sometimes they get pretty big, and you can remove them. But it's unless you do a biopsy and take tissue out, you're not sure. These malignant ones metastasize, they just flake off, break off, and so what they'll do is jump. They'll jump into a blood vessel and then catch a ride, and then they pop out somewhere else. It could be through the blood, but it can also be through the lymphatic vessels. So that's again why one of the first areas they look for cancer is in lymph nodes, because lymph nodes are filters and they trap that kind of stuff. But you can destroy them. We can destroy them. And you probably are destroying them right as I speak. Little weird cells that are misbehaving. And there's a T cell that's small, it's like David and Goliath or something. A little small cell blowing up a big cell, a cancer cell. Some cells are big, some are small. And those are the holes with perforating. That would be a killer T cell perforating the cancer cell. And once you pop it, it can't divide, it can't grow, so the tumor won't spread. Question?
Can a non-malignant tumor turn into malignant if you let it stay there and grow?
I'm sorry, I couldn't hear the whole thing.
Can the non-malignant tumor turn into a malignant one if you let it grow and stay there?
Well, can they shift? That's possible. It's always possible. That's why, I mean, I think probably just about everybody in here has been affected by cancer one way or another, either directly or family or friend. I mean, everybody has stories about cancer. And you know that if somebody's going through cancer treatment, it's never over. You've got to keep checking, just constant checkups, because you just never know if the tumor will jump and suddenly become malignant or something, or start to spread. So, yeah, it's constant vigilance. Yeah.
So, in the picture right there, how is the T cell lysing the cancer cell? Is it punching holes in it or defense?
Yes. How does the T cell lyse the cell? We went back, remember perforin? It's pretty similar to complement fixation or MAC, where you drill holes, perforate by lining, putting proteins in there that pop holes into it. And that's what it did. That's what T cell lysis. That's the whole idea of lysis. T cells do the hand-to-hand combat. They blow up, they go right next to the cell and blow up. Okay, that's all I'm going to say about cancer. I mean, do an entire course on cancer, spend your entire life studying cancer, because it's very complicated, and it's a lot of medicine involved there too. But I also want to look at tissue transplants because this involves the immune system. And so there's different ways you can do transplants. You know, whether liver transplants, kidney transplants, heart transplants, all kinds of different things. So it could be a skin transplant. You've got a serious burn injury. So ideally, the easiest thing to do is self-to-self. Why would that be the best way to do a transplant? There's no immune system problem. It's self-to-self. Your body already recognizes it. There should be no tissue rejection. The next best thing you can do is an identical twin. So, well, because you're genetically identical. So therefore, hopefully your proteins are the same. Now there can be differences between identical twins because you start off genetically identical, but the environment can influence certain things and cause changes over a lifetime. But the genes initially start, they're all the same. So, so isograft is an identical twin from identical. So you go from self, you go identical twin to identical twin. Allo means other. Graft. So between genetically different people, that's the most common. So you take a person and the neighbor next to you donates their kidney to you. They're different genetics. That means they're going to have different MHC. They can do a bunch of testing though to find somebody who's more similar to you to get it as close as possible. There's always the risk of tissue rejection because you have different immune systems. You have different proteins and receptors on your cells. And then the xenograft is you take from another animal. Like you're taking from a pig and then take that tissue and put it into a human. And of course, there's even more serious tissue rejection. But all of these are possible and they're all very good. That's all I'm going to say about tissue transplant. I'm going to work about allergies. Cover your mouth. Okay. So, and the thing is, I just see, so this is what you're supposed to do. You gotta get close. It's like that. You do this, and then you go open the door, and then everybody opens the door after you. They all, So, if you work in a hospital, it's already full of germs. Don't add more. And I have to say, just watching, sometimes doctors and nurses are the worst. Maybe you get a little bit lax about it because you're around all the time. But every time you're meeting so many people and they're all sick, the germs keep going around, and that's why a lot of these super germs end up developing also in hospitals because of the combination of high frequency of germs and high frequency of antibiotics. They're not fully used, and so you go to the hospital and you get like some super germ. It's really hard to fight. But allergies, sort of like catching the flu or something, except that it's pollen. Oh, it could be dust mites, it could be dog dander, cat dander, it could be anything. So you get a little scratch test to figure out what you're allergic to, and if it swells up, that means you've got a histamine reaction. Has anybody ever had a scratch test? Okay, you do a little scratch, they prick your back or your arm, and they do a difference between, it might be just all trees, tree pollen, or something, it might be grasses, it might be animals. So you do that, and anything that swells up fast, that's histamine being released by basophils, which are white blood cells. So histamine is definitely a thing. So it'll make you sneeze. Your body's trying to push it out of the water. It could be mold spores, the little small microscopic things. Any allergy. An allergic response is triggered by anything, chemical, pollen, insect bites, you name it. Some people are more allergic to others. A lot of that's just genetic. And so plasma cells make antibodies initially, and they could be sensitized. I still don't understand this. You can become sensitized or you can become desensitized. I know so many people that suddenly randomly become allergic to almonds, or crab, or shrimp, or peanut butter or something. All of a sudden, you're 30 years old or 40 years old, and then sometimes happens when you're 15 years old. How many of you become allergic to something suddenly? Anybody? Just a couple of you? I mean, but there are a lot of allergies, and I don't know if there's more allergies or we're just realizing them more. Some people think there are actually physically more allergies now, and we're trying to figure out why. But in any case, The allergies are driven by antibodies. The antibodies are shaped to pick up the pollen, which is good. They're supposed to fight the pollen. But if you overreact, then that's not so good. So it's antibody mediated, and they're driven by mast cells. Mast cells are basically basophils that live in tissues. And so if the pollen fits on the little receptors that are on the basophil will release its granules, which are the dark blue staining things you saw in the lab practical yesterday, the dark blue thing. Those are histamine. Histamine is a chemical that causes the swelling, runny eyes, runny nose, coughing, and all kinds of swelling. It will cause your throat to swell up, cause asthma. So there are a variety of them, and I won't typically don't go through all of these on the test, but one I want to show is anaphylactic shock. This is a very serious allergy, and this happens like with bee stings, but it could be a food allergy. And they're very fast. You might get this, if you are allergic to peanuts and you eat a peanut, you might go into an asthma attack immediately. And if you don't get that corrected, possibly you could die, or you need to get an adrenaline injection really quickly, an EpiPen, epinephrine. Epinephrine is adrenaline. That dilates the airways. That's the first thing you do if somebody gets into an immediate attack. But anaphylactic shock means system-wide, systemic, means everywhere in your body. Tips of your fingers, toes, your lungs, everywhere. So it's a serious allergic reaction. Usually they're localized. Like if you get poison ivy, or a normal person has a normal bee sting that swells up and hurts right there. It doesn't hurt your whole body in there. So you should know what anaphylactic shock is. There's subacute types of allergies. Blood reaction, normal asthma, if you're allergic to dogs, maybe a couple hours in the house with a dog.